Today, we will start with a new unit on invertebrate diversity. Most high school classes cover this topic in a day, but we will spend 5 weeks on it because I believe it is a much more exciting way to review ecology, evolution, and physiology. You all know what dragonflies and sand dollars look like, but you probably don't know why they look the way they do and why they are important to the planet. Hopefully, this will give you a greater appreciation for how amazing nature is and why it is important to protect it. Here's a look at the phyla we will be covering on a phylogeny, with some of the most important traits they possess. We will begin by focusing on the first animals that branched off, meaning the ones that evolved first. Which phylum branched off first on this tree? Remember that animals evolved from unicellular protists. They are eukaryotes, like protists, and really have all the same features, except they are multicellular. How did multicellular evolve in animals? There are these protists called choanoflagellates that are still around today that we think are closely related to the ancestor of all animals. They kind of look like sperm cells with short tails and a collar around their flagellum. The flagellum is whipped around to create a current, like the blades of a fan, sucking in water and food particles that get trapped in the comb. This is how they catch their food. And sometimes a handful of these choanoflagellates form a colony, where they share resources and may even have a stalk that connects them to the ground. So, just like plants, colonies form before being truly multicellular.
For 2.5 billion years, single-celled organisms dominated the Earth. They were minuscule creatures, completely enclosed in a fragile membrane. These were living, pulsing beings, but they were not animals. Somehow, cells developed a language that allowed them to work together. When they did, it was a turning point for life on Earth. The very first animal had begun to take shape. Ever since Darwin put forth his theory of evolution, scientists have puzzled over animal origins. At long last, scientists think they have found a creature that links us to a moment in deep time, to the first animals of Earth. It is a creature that is somewhat alien in its appearance, a being without a nervous system, muscles, limbs, or a brain. Its body is defined by a loose assemblage of cells. It is the ancient sponge.
Today, our focus is on sponges. Yes, sponges are alive, and yes, they are animals.
When we think of animals, we conjure up something entirely different. Animals move. They feed themselves. They reproduce and care for their young. In comparison, a sponge seems lifeless. If it is an animal, it certainly conceals its true nature.
They are very simple, and hopefully you will see how we go from a single-celled amoeba to much more complicated whales and octopi. Many sponge cells looked identical to choanoflagellates. We call these cells choanocytes. Dye has been injected at the bottom of this sponge. What do you see the sponge doing?
These ghostly exhalations are proof that the sponge is actively pumping. With no mouth, it sucks water directly through its sponge-like body walls, which are shot through with millions of narrow canals and tiny chambers. How does it do it?
You should have seen that the sponge was using its choanocytes to generate a current. Water enters through pores on the side of the sponge and exits through an opening in the top called the osculum. Hopefully, you are taking notes on all these new vocabulary words. Sponges belong to their own phylum, Porifera. This phylum name is easy to remember because sponges are filled with pores. They use these pores to suck in floating organic matter called detritus, and marine organisms called plankton to eat them. This is called filter feeding.
A sponge is actually a fantastic pump, which requires an incredible coordination of cells to function. It's a living filtering machine. It lives by soaking in water and filtering out food particles.
These seemingly motionless creatures are working extremely hard in order to feed.
Imagine that to get an ounce of food, a sponge has to pump over a ton of water through itself. Now, can you imagine doing that yourself? Drinking a ton of water to get an ounce of food to your body?
Drifting along with countless single-celled organisms, we feel the tug of a current. The walls themselves are treacherous. They can simply engulf their unfortunate prey. As we are drawn through ever narrower passages, the current slows until we burst into one of the sponge's millions of tiny beating hearts. These are choanocytes, cells whose whip-like flagellae powers the pump. The flow quickens again as it whisks us away through the labyrinth of canals that lead out into the sponge's central cavity. What once seemed barely alive is actually a living animal, more complex than we could have imagined.
There are 9,000 described species of sponges, and most live in the ocean, but 150 or so live in freshwater.
From the frigid waters of the poles to the warm seas of the tropics.
They can grow to look like vases or flat on rocks. All are sessile as adults, meaning they cannot walk around. They come in a variety of colors, but lack a great deal of physical diversity. Yes, the sponge you used to do the dishes was inspired by the sea sponge, but modern sponges are synthetic, made from polyester and not the skeleton of once living sponges. Sponges have no symmetry. They're really just a jumble of cells with no real organization. The cells do not develop into germ layers, and they don't have true tissue because although they have several different types of cells, they don't form an organ with a particular function. Each cell is really working on its own, but benefits from being with others. How do you think a sponge manages to have sexual reproduction? Male sponges produce sperm and females produce eggs. But how do the sperm cells reach the egg?
The sponge's pumping does more than satisfy its hunger for food. In fact, its sex life depends on pumping as well. 500 million years ago, these were the very first animals to reproduce sexually, the first to combine sperm and egg to create offspring. Set adrift, some sperm will be lucky enough to enter another sponge of the same species. If the sperm are truly fortunate, they will fertilize the eggs within.
Once the egg is fertilized, it becomes a spiky, free-living larva that settles down and grows into an adult sponge. Some sponges can also undergo asexual reproduction through fragmentation. Because they lack tissue, any cell can get cut off and create a new sponge. In fact, you can put a sponge in a blender and any leftover cells that are not destroyed will grow into new sponges.
We need sponges to study in the lab, and so we have to collect specimens. But we need to just cut a small section to be examined, and because it doesn't have a nervous system, we are not really hurting it. In time, the cut area will grow again as if nothing had happened to it.
All animals rely on cell-to-cell communication. But Diaz and others have been able to show that sponge cells have a unique way of working together.
Cell-to-cell communication is very important for all organisms. But in sponges, it's uniquely important because in sponges, cells carry out all the functions that other organisms are taken care of by specific organs like the heart or the nervous system or the skeletal system or the blood. So sponge cells have many responsibilities to perform.
Unlike most of our cells, those of sponges retain a special freedom. They can continuously reinvent themselves, performing some truly miraculous feats. If a sponge is passed through a sieve, its cells are separated. But somehow the cells still recognize each other. Over several hours, they begin to come together, building many new sponges. No other plant or animal can resurrect itself this way.
Sponges have three main types of cells that look different and serve different purposes. Choanocytes I have already mentioned. These are the ones that are used to gather food, oxygen, and gametes. The outside of the sponge is covered in flat cells called pinacocytes that form a skin around the sponge that supports and protects the inner cells. The third type of cell are amoebocytes, and they look and act exactly like amoebas. They wander around the body of the sponge, taking food from the choanocytes, digesting it, and providing nutrients to the cells that need it. In this way, they function like our blood cells. They can also develop into pinacocytes or choanocytes and are therefore a type of stem cell. Lastly, they produce and secrete three different types of fibers. Fibers are a strong material that are difficult to digest. The first one the sponge produces are called spicules. These hard, spiky objects made of silica or calcium carbonate make predators think twice about eating a sponge.
Spicules are microscopic structures that help give a sponge its rigidity and form.
Spicules are an incredible tool for us today to identify most of the sponges that are living in the oceans.
For most sponges, the glassy spicules act as a kind of fingerprint. Magnified, their dazzling array of shapes and sizes are as beautiful as they are unearthly.
The second fiber is called spongin, which creates the skeleton of the sponge. Lastly, the amoebocytes produce collagen, which glues the cells and fibers together to create a multicellular organism, the first animal.